Hello, everyone. This is a momentous occasion. There are only a few things that I absolutely love when it comes to biology, and today you're going to meet one of them. Oh, you'll know it when you see it. I was so floored by it. I literally didn't even know what to do when I first heard about it. And then one of my former students, she went to uh, Baylor University College of Medicine, and she said she had a professor that was one day lecturing and said that he was the one who discovered it with a group of other people from other universities. Okay. Anyway, here we are in chapter 17. We're going to do 17.3, which is all about post-transcriptional processing of the messenger RNA. I'll get into that detail. It's what gives eukaryotic cells, I think, their character and their ability to basically be better than prokaryotic cells, more efficient, burn food better, and then allows the organisms that they make when they group together to be more complicated and complex and do more complicated things. So, 17.4 is all about translation. We're not going to get into the nuts and bolts of actual translation today. We're just going to set it up. We're going to talk about tRNA and the recruitment of the ribosome and things like that. But first, eukaryotic cells modify RNA after transcription. Go ahead, press pause, and tell your neighbor what you think happens there. What do you think happens? Well, let's back up. Do this. Maybe this will be one of the last times you ever do this. Do this by heart, please. Fill this out. Press pause. Fill it out before I even do. Remember that transcription is the process by which the message on DNA gets turned into what we call RNA. Then translation is the process by which that message is translated into a sequence of amino acids. Then those get folded appropriately into a functioning protein that actually does something in the body. Don't know why I did that either. When it comes to the mRNA processing, which is what I'm trying to get you excited about here, here's the first thing that happens. 5' cap. That just means you have this mRNA transcript that comes from the RNA polymerase in the process of transcription in the nucleus, and a 5' cap gets put on it. What's interesting is we've only realized recently what this 5' cap is, and it's a G3P molecule. Have you seen this before? Remember, it's the intermediate in the dark reaction or the Calvin cycle of photosynthesis. We see the same molecule, glyceraldehyde 3-phosphate. I don't know why, but I think it's pretty cool that it shows up here again. That does have a function to allow the mRNA to attach to the ribosome more effectively once the mRNA goes out into the cytoplasm. So the 5' end receives a nucleotide cap. There it is, symbolized by that little bit of yellow on the surface of the mRNA there. And the 3' end gets a poly A tail. So if you recall, the termination sequence on the DNA was TTA, TTT. And so that's going to cause mRNA to be made that's AAUAA. But the point is there's a lot of A's here. And the idea is that it's about 50 to 250 nucleotides long, depending on how long the cell wants the protein to be made, meaning the translation of this mRNA to occur in the cytoplasm. What happens is that you can't, and I've said this before, we have to regulate. So you can't just let proteins be made and just made. So when an RNA goes out into the cytoplasm, it can't just sit there and be translated forever. It has to be broken down. And so the moment that an mRNA gets out into the cytoplasm, the ribosomes attach, translation begins to make amino acids to turn into a protein, degradation begins. So there's these hydrolytic enzymes out there, and all that means is that they use water to cut, and they just basically start chewing up the mRNA. But the longer you have this nonsensical tail on there, the longer that the mRNA will exist in the cytoplasm and the more protein will be made. How do they know which proteins to put a 50 nucleotide tail on or a 250 nucleotide tail on? I don't know. Isn't it wonderful? Mysteries of life. These modifications have several functions. They facilitate the export of the mRNA to the cytoplasm. They protect the mRNA. In the case of this guy, let me show you what these hydrolytic enzymes look like. They basically find the three prime end, and then they just start chewing off nucleotides in the mRNA. This is called a dicer. It's just a protein that cuts mRNA. Basically, that's its job. Using water, it breaks those bonds. It's pretty exciting. I hope I haven't given you the fuel for any nightmares here, but we got to regulate. We got to break down the mRNA. We can't be making proteins forever, only when we need them. That speeds up. Okay, sweet. So these also help the ribosomes attach to the 5' end for translation. So we're going to watch one more movie.
Before mRNA can be used by ribosomes as a template for building proteins, it must first be processed. Key steps are the addition of a methylated cap and a polyatomic tail. Involved in processing are RNA polymerase, cleavage factors, and poly-A polymerase. Processing of mRNA begins with transcription. Soon after RNA polymerase begins transcription, a methylated cap is added to the 5' end. Transcription then continues to completion. Following completion, RNA polymerase releases the capped strand of pre-mRNA. Specific nucleotide sequences in the mRNA are bound by cleavage factors. The 3' end of the mRNA is next moved into the correct configuration for cleavage. Stabilizing factors are then added to the complex. Poly-A polymerase now binds to the mRNA and cleaves the 3' end. The complex begins to dissociate and the cleaved 3' end quickly degrades. Poly-A polymerase now synthesizes the polyadenylated tail by adding adenine residues to the cleavage site. Additional proteins then bind to the tail, increasing the rate at which it grows. When the tail reaches its full length, the poly-A polymerase is signaled to stop adding residues, and the polyadenylation process is complete. The processed mRNA is now ready to undergo splicing in preparation for translation.
All right, she hinted at it. Splicing. We got to cut this thing. Right here. mRNA splicing. So here's the idea. One of the very few things that makes us different than these prokaryotic cells, the eukaryotic cells that we have, is this processing. We got this, no problem. But what about the cutting? Well, we have about 20 to 23,000 genes in our body, in the DNA of each of our cells. However, we make over 1 million different proteins. I did the math. It's like 43 or 46 times more proteins than we actually have genes to create those proteins. If you believe that this is a linear thing, then we should only make 23,000 proteins, right? How do we know how to make 40 times more proteins than we actually have a message for? Spliceosomes. Spliceosomes are the things that do the mRNA splicing. So you can see here on the cartoon that the mRNA is much smaller, the mature mRNA before it goes out to the cytoplasm, much shorter than the pre-mRNA. Well, these two organisms, the nematode worm on the right and then a sleepy toddler on the left, have the same number of genes, about 23,000. But it's what you do with those genes that allows you to increase your complexity. Well, this is what science has found in the last 20 years. So there are segments of the mRNA which are removed. So these are called introns, and there's usually about, it's called a SNRP. It's a sequence of letters about 150 nucleotides long at the beginning of each intron, and that's going to allow these spliceosomes to cut at those places. So they're going to cut there, they're going to cut there, and what you see here is that they're going to join the exons. So then the exons are sequences of mRNA letters which are then put together. They're spliced together by the spliceosomes. So the spliceosomes cut at certain places, and then they put certain sequences of the mRNA together. But here is the beauty of it. The mRNA length is reduced by about 95.5% on average between the mRNA transcript here and the actual mature mRNA that goes out to the cytoplasm. Here's the question. And when I die and I'm chilling out with Abe Lincoln and Martin Luther King, and I see the light, this is the question that I'm going to ask. How do spliceosomes know how to do this? And this is where their genius lies. Instead of just putting, let's imagine that these are the places that a spliceosome cuts. Instead of putting A next to B, next to C, next to D, the spliceosome could rearrange this. They could put A next to F, next to C, next to D. Or they could put D next to A, next to F, next to C. Use the same letters, that was weird. But what you need to understand is that if you take a sequence of letters and you cut some out, it shifts everything over. And so even if I had 1,000 letters in a sequence of mRNA here, and I just took out one, let's say with a spliceosome, and everything shifted over, now instead of this being like this, then the next letter that's here will now be responsible for the next amino acid in a sequence. And the idea is that it gets crazy, the permutations here. If you think about how complex this is, how do the spliceosomes with no brain know how to rearrange these things so that we can get 40 times more protein? Your book will say each DNA is responsible for about 8 different protein types. But I like to subscribe to the number 40, that they're rearranged to make about 40 different types from one segment of letters on DNA originally. You can tell that this gets me excited. Spliceosomes, they're amazing. I don't know how they do this. I don't know how they choose where to cut and how they know what to put together. This is what I was just suggesting here. If you just shift one letter over, everything in terms of amino acids changes. That means the folding changes, that means the shape changes of the protein, the resultant protein, and that means the function of the resultant protein changes. It all changes. It blows my mind. They are spliceosomes. They consist of a variety of proteins and several small ribonucleoproteins called SNRPs that recognize the splice sites. I have a couple movies I'm going to show you here. In fact, you just need to write this on the test and you're guaranteed extra points. They say no extra credit at Battle Mountain High School. That's wrong. Petition my soft side, which is spliceosomes. I love Golgi bodies also.
Here we see an RNA molecule with a single intron. Several signals exist within the intron that are used in the splicing process. From the 5' end of the intron, these are GU, the A branch site, a pyrimidine-rich region, and the 3' AG. The GU and AG sequences define the beginning and end of the intron. Splicing is mediated by the spliceosome, which consists of several protein RNA complexes. The first step involves 2 complexes that bind near the GU sequence. The RNA is then looped, and 3 other protein RNA complexes bind. This final complex then undergoes a conformational change. Introns are non-coding RNA sequences that must be removed before translation. The process of removing the intron is called splicing. The intron is then cleaved at the 5' GU sequence and forms a lariat at the A branch site. The 3' end of the intron is next cleaved at the AG sequence, and the 2 exons are ligated together. As the spliced mRNA is released from the spliceosome, the intron debranches and is then degraded.
As DNA is transcribed into RNA, it needs to be edited to remove non-coding regions or introns shown in green. This editing process is called splicing, which involves removing the introns, leaving only the yellow protein coding regions called exons. RNA splicing begins with assembly of helper proteins at the intron-exon borders. These splicing factors act as beacons to guide small nuclear viral proteins to form a splicing machine called the spliceosome. The animation is showing this happening in real time. The spliceosome then brings the exons on either side of the intron very close together, ready to be cut. One end of the intron is cut and folded back on itself to join and form a loop. The spliceosome then cuts the RNA to release the loop and join the 2 exons together. The edited RNA and intron are released and the spliceosome disassembles. This process is repeated for every intron in the RNA. Numerous spliceosomes, shown here in purple, assemble along the RNA. Each spliceosome removes 1 intron, releasing the loop before disassembling. In this example, 3 introns are removed from the RNA to leave the complete instructions for a protein.
Couldn't you just watch it all day? I often wonder if I like people or not. I mean, I love young people and I'm excited about their futures and trying to help them. I love plants, like my family, and I love spliceosomes. Oh my gosh. I don't know how they work. I'm completely flummoxed by them, and I'm just fascinated by them. So I hope you are too. This is one of the mysteries of the great mysteries of life, in addition to neurology and I have lots of other things. Please press pause and go back over what happens in mRNA processing. If you want as much extra credit as I will absolutely give, you should write down that these 3 things rock on the next test. They certainly do. 10 years from now, you won't remember me or this classroom or who you sat next to, but I hope that you remember that you need to shop the perimeter, get a cutting board, and that these things rock. And that's it. Then, I'll be satisfied. You won't remember me. I'll pass you on the street. You'll be like, and that's okay. As long as you remember this, please. Well, let's translate that mRNA. Now we're out in the ribosome. All this post-transcriptional processing has occurred. Translation is the RNA-directed synthesis of this polypeptide, these sequences of amino acids in a row. Here it is, translation.
When the RNA copy is complete, it snakes out into the outer part of the cell. Then, in a dazzling display of choreography, all the components of a molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. Each transfer molecule carries a 3-letter code that is matched with the RNA in the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecules. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA.
Okay, so here are the players. Ribosome, there's going to be 2 subunits. This is made up of rRNA and proteins. Literally, it's RNA, which is just, instead of being a message like mRNA, it's just folded with some proteins. It's become this thing that's going to recruit the correct tRNA to come in and land on a certain grouping of letters on the mRNA, and then bring an amino acid into a sequence. And we'll get into all that detail next time at the end of 17.4. But we just need to kind of go over the big picture here and then transfer RNAs. Those are the kind of the soup can things that you see up here with the purple thing attached to them. Those are amino acids that are attached to the tRNA. So go ahead, predict how this is going to work. This is translation. Say it out loud right now. Press pause, please. Well, a cell translates an mRNA message into a protein. Sorry, that's very clunky wording there from the book's publisher. With the help of transfer RNA, but I'm responsible for what I put on the screen, so that's my fault. So here's the tRNAs. Let's talk about tRNAs. So tRNAs are like 80 nucleotides long. They're basically exactly the same as one another. There's 20 different types. The only 2 things that they do differently is that they have something way down there at the bottom called the anticodon, which differs. It's like 30 letters in from the 5' end, and that's going to be different on all 20 different tRNAs. And then what they do with the amino acid attachment site, way up there at the top at the 3' end, the ACC, that's where a specific amino acid will attach, and it's different for all 20 transfer RNAs because there's 20 different amino acids. The tRNA molecule consists of a single RNA strand, you can see, which is folded back on itself in kind of this cross or T shape. It's 80 nucleotides long. It's very, very consistent. You can see those hydrogen bonds that have formed there. Remember, RNA is single-stranded, but it can fold into these crazy shapes, and we're about to learn more about that, and then make those hydrogen bonds. So RNA can actually create very rigid structures when you get a ton of this hydrogen bonding here. Molecules of tRNA are not identical. I already told you how they differ. And why do they differ? Well, they differ because the different ones grab onto different amino acids, and that's important. Each carries a specific amino acid on one end, way up there at the top. ACC is always the same. And then each has an anticodon on the other end. So the anticodon is just going to be the opposite of the codon. So this is actually what's going to bind to the mRNA, this anticodon. So the question becomes here, here's our little activity. What would the amino acid be on the tRNA? How would you figure that out? Press pause and try to figure it out. So remember, the mRNA is going to be read in a 5-to-3 direction. And so if you have an anticodon, I'm looking at the top there where the ACC is at the 3' end. So if I go down then it's A, G. And so if that's the anticodon, what would be the codon on the mRNA that's this thing is sticking to momentarily? It would be 5-to-3. Let's read it. It would be U, U, C. Do you see that? Because the A is going to correspond, I'm at the anticodon. The A is going to correspond to a U, and a C going from 5 to 3. Now, if we look at this a codon chart on the far left side, it says first mRNA base, and that's a U, right? So let's go over and find where the U's are. So let's go back. Let's make sure we have this right. It's going to be U, U, C. So it's going to be U. Where are the U's? Okay, find the U's. Okay, great. And then find the second U, U, U. Okay, I see that. And then C. And which amino acid, well, I'm going to go back. Which amino acid would this anticodon having tRNA have on its amino acid attachment site way up there at the top? And if you said that the word phenylalanine or PHE, you are correct. This is kind of what it looks like in cartoon form. I'll show you a movie on this in a second. We like to simplify it down to the soup can here. It's just much simpler to not show all that complexity. Due to hydrogen bonds, the tRNA twists and folds into a three-dimensional molecule.
All tRNAs have a characteristic L shape, with an amino acid attached to the 3' end at the tip of the shorter arm. The anticodon loop is positioned at the opposing end of the molecule and contains the anticodon base triplet. The amino acid, a phenylalanine in this case, is covalently attached to a conserved sequence, CCA, that is common to the 3' terminus of all tRNAs. The anticodon is comprised of 3 nucleotides complementary to the codon in the mRNA. The bases are exposed and are thus freely accessible for base pairing during protein synthesis. In this example, the anticodon sequence is GAA, which would match a UUC codon on a messenger RNA, specifying phenylalanine.
Yes, we got it right, phenylalanine, sweet. So here's the question. So you got the mRNA, you got the ribosomes. I get it. The ribosomes are going to recruit the correct tRNA with an amino acid attached to it to come down and lock momentarily onto the mRNA to put that amino acid into the growing sequence of amino acids, the polypeptide chain that we're making over there in the central dogma little formula that we had. And so the question becomes, then what happens? Well, so the tRNA has now lost its amino acid. So what happens to the tRNA? And you probably guessed, similar to what happens to ATP, is it's going to get recycled, right? We're going to use it again. And so how does that happen? And your book goes into this nice paragraph or two about how a naked tRNA gets recharged with the appropriate amino acid. So accurate translation requires 2 steps: a correct match between a transfer RNA and an amino acid done by this enzyme, aminoacyl tRNA synthetase enzyme. You can imagine there that we have this enzyme, and it's going to recruit an appropriate amino acid in there with the help of ATP to recharge, you can see it, to recharge the transfer RNA. Now, I used to think that there were 61 different, because there aren't 3 tRNAs for stop codons. There's just nothing, it's like a protein that blows up the whole translation process at the end of it. But there are basically, if you look at that codon chart, there are 61 other codon sequences which can cause a tRNA to come in with an amino acid attached to it, including the start codon, which is AUG, which is methionine. So I used to think that there were 61 different aminoacyl tRNA synthetase enzymes, and they would latch in with their tRNA. You can kind of see it in the middle of that picture there, and then the correct amino acid was already in there, and then fused the two using ATP. And now all of a sudden we have this charged tRNA which can go into the cytoplasm, be recruited by the next ribosome, add their amino acid into sequence. Well, I had a student this last year who got a 5 on the test and who's way smarter than I am. She said, what if there's only one aminoacyl tRNA synthetase enzyme? And in fact, it recruits the naked tRNA first, and the codon on the bottom, the only thing that's different on any tRNA, those 3 letters, on the anticodon on the bottom, that causes a conformational change in the protein so that the site way up there, the site where the amino acid goes in, changes shape, and therefore one of the 20 different amino acids is then able to get into that site and then fuses to the tRNA that was once naked and now it's charged again and it's ready to go. I think it's genius, and I think it's the way that it happens. Another mystery of biology. I don't know the answer. Probably we could look it up. Probably
for a molecule.
So what we're seeing here is we're seeing a bunch of these tRNAs floating in the cytoplasm. These guys are charged. They have amino acids on them. What the ribosome does is it grabs onto the mRNA and it basically pulls the mRNA through like a tape, 5 to 3. So it starts at the 5' end, goes to the poly A tail at the 3' end. As it's doing that, the ribosome somehow, I don't know how it does it, but it recruits the correct tRNA once it reads the 3 letters on the codon that you can see down there right in the middle, brings the correct tRNA in. The only job of the tRNA is to get in there for a moment, add its amino acid to that growing polypeptide chain. We'll get into way more of this detail over the next day or so in 17.4, but I just want you to see it one more time. It's incredible, the complexity here and how everything has to be done right. And this is happening in every cell of your body. And who figured this out? And how does it even work? I think it's amazing. I really do. The correct match between the tRNA anticodon and an mRNA codon is super important, obviously. We've got a problem when it comes to this. There's only 20 different amino acids that need to be delivered, but there's 61 different sequences of codons that account for those 20 amino acids. So we have a redundancy, I guess is what I'm trying to say. There are different codon sequences that you can see on this codon chart that cause the same amino acid to be put in a sequence because even though it's causing a different tRNA to come in, it's the same amino acid up at the tippy top of it. Why I call that a problem is scientists didn't know what that was for or how that worked or why we had evolved to have that for a long period of time. And this is called wobble. What's fascinating about this is that you can see that the variation between the nucleotides is only in the 3rd letter of the codon. Do you see that? Let's look at serine, way up there at the top, SER. You can see it's UCU on the mRNA codon, which will code for a tRNA, which has a serine amino acid attached to it, or a UCC or a UCA or a UCG. First 2 letters are conserved. Last letter can be a little bit variable in a lot of these. And one of them, leucine, if you look at that on the left side, there's 6 different codon sequences that cause the same amino acid, leucine, to be put in sequence. That probably suggests that leucine is pretty important. Why is this important, this thing called wobble? Well, what's cool about it is that what it means is that you could have a mutation on DNA. Let's say you're laying out at a single tree pool, UV light hits your arm, causes a mutation in the nucleus of a cell, mutates a T into a C on the DNA. You don't really know it, but that cell is going to then, it's going to do a lot of things. It's going to make a bunch of proteins. But the point is that the mRNA that's made from that mutated DNA now has a different letter where there once was one letter, now there's a different one. And the question is, that's fine as long as the protein that needs to be made is correct, or it's the same protein as usual, so that cell can do the same jobs as it's always done. And so why this is important is because you can have that mutation in your DNA and you still get the same amino acid placed in a sequence and you still get the same protein made. And you're none the wiser. You have no idea that there was actually a DNA mutation that occurred. And that is why scientists believe we have evolved to have this redundant system and why it is so complex and beautiful. Press pause and take a moment and go over what you know so far about translation. Thanks. See you next time.